Hello and welcome everyone to IRIS Biotech's 10th online workshop. True to our slogan, Empowering Peptide Innovation, we are keen on knowledge exchange and getting in contact with scientists all over the world. Therefore, we started this workshop series 2 years ago. Today, our focus is on cell-free bioproduction of proteins and peptides. Our guest speaker, Dr. Stefan Kubik, will present a fascinating method portfolio about protein production for therapy, diagnostics, and biotechnological applications. My name is Raimund Meyer. I am working as a COO at IRIS Biotech, and today I will guide you through this online workshop. If you wish to announce your participation on social media, please feel free to use the hashtag IRIS Workshop Series, which is also displayed right now in the Microsoft Teams chat. To have a pleasant experience for all participants, as always, I would like to begin with some technical guidelines. To ensure the best possible audio and video quality, all participants are automatically muted and your video camera is switched off. This live event is recorded, thus if you enter late or miss anything, please check on the bottom left side of your Teams screen if you are watching the recording or if you are live, which is shown by the red broadcasting symbol. You have the possibility to pause the video, rewind, or jump back to the live session. In addition, the recording of the workshop will be uploaded on our homepage afterwards. Questions can be submitted by using the Q&A chat of Microsoft Teams. You can enter your name or submit your questions anonymously. We will frequently check the questions, publish them, and submit them to the respective speaker. If you have any technical queries, you can also submit them in this Q&A chat. Our technical assistant will then connect with you and try to solve the problem. Initially, the questions are just visible for the organizers and will then be published to the audience. So don't worry if your submitted question does not appear on the chat screen immediately. I would now like to give you an overview on today's agenda. We will start with a short company and topic introduction by our Chief Scientific Officer, Dr. Thomas Bruckdorfer, and afterwards our external speaker, Dr. Stefan Kubik, will present to you his superior methods for cell-free protein production. As mentioned, questions will be collected throughout all talks in the Q&A chat and are forwarded to the speakers. Of course, you can also get in contact via email or phone call after our workshop. One last note from my side, if you accidentally close your Microsoft Teams window, don't panic, as this workshop is also available as recording, you won't miss anything. Just relog in and view the recording. You can also play this recording at elevated speed until you catch up with the live event again. So after this technical intro, let's continue with the first agenda point, a short introduction to our company by our CSO Thomas Bruckdorfer. Thomas studied chemistry and obtained his PhD degree at the University of Erlangen. He broadened his education by an MBA study in management and distribution of complex technical projects and systems, and is now a co-owner of IRIS Biotech since 2002, serving as Chief Scientific Officer and Vice President Business Development. Thomas, thank you for your time. Please go ahead with your presentation.
Yeah, thank you, Raimund, for your kind introduction, and also welcome from my side to this workshop. With the following slides, I want to give you a brief introduction to our company, IRIS Biotech, as well as to the topic, which is cell-free production of proteins. IRIS Biotech has been founded 22 years ago in 2001. And we are located in Marktredwitz, which is in Bavaria in Germany, close to the border to the Czech Republic. Due to the growing demand of customized products, we have started a production facility, IRIS Biotech Laboratories, in 2013. This is located in Willstedt, in the southeast of Germany, close to Strasbourg. Our portfolio today consists of more than 6,000 products, which are produced in our own laboratories and by some 15 selected manufacturing partners. We select actually very carefully with whom we work. which results in a very low complaint rate, which is consistently below 0.5% only. And we do not only want to give you facts and figures, but also would like to share with you some impressions of our location. We are located in a rural area in the mountains of northeast Bavaria. And this is not only a nice location, it's actually a hotspot of technology-driven small and middle-sized companies just like us. Here you see a picture of our company building in which we moved in 2018. So we understand ourselves as a qualified supplier and competent consultant, and want to support and accompany our clients during design and execution of their projects, no matter whether it is in research scale, further pilot, or commercial production. Our main markets are still reagents and building blocks for the design of peptides and its modifications. We also provide polymers like PEGs and polyamino acids, which are used, for example, as components in drug delivery applications. All is rounded up by our custom synthesis service division. Since a couple of years, we are developing permanent and particularly self-immolative linkers, which find more and more interest. And this actually brings us also to the topic of the webinar of today, which is cell-free production of proteins. Due to our slogan, empowering peptide innovation, we are keen to interact with innovators of sophisticated technologies and make such technologies accessible to a broad audience and the market, basically. This is also the reason for starting of our webinar series, having an information platform available which explains latest technologies and describes benefits and possibilities of latest inventions and how they can be used now by everyone. And this brings us now to the topic of today, which is how cell-free protein synthesis works and how bioorthogonal chemistry can be carried out in this context. It's a real pleasure for me to have Dr. Stefan Kubik with us as a speaker, explaining all details of this specific technology. Some key figures up front. Cell-free protein synthesis is an in vivo production system which can produce all kinds of biomolecules, such as proteins, antibodies, certain antibody formats. And with this technology, synthesis of compounds can be carried out which could not be synthesized before, such as toxic proteins, for example, membrane proteins or antibodies, which carry non-canonical amino acid at very specific positions, which can then further be used for attachment of linkers and payloads. Furthermore, it's an open system which allows fast initial production of small quantities and later on also a fast scale up to commercial quantities. It is also a true synergy to our own capabilities at Iris Biotech in the field of linkerology, as it prepares biomolecules for linker and cargo attachment. In combination with Dr. Kubik's capabilities, we are closing a gap and are now capable to provide any kind of carrier-cargo conjugate. So with these words, I want to close and hand over and look forward to hearing more details.
Yeah, Thomas, thank you very much for the kind introduction. For technical reasons, I will switch off the camera and I will switch on the camera immediately after the presentation.
Okay, so if I may shortly interfere, because I would like to give some introduction to our guest speaker. Stefan Kubik achieved his PhD in molecular biology and physiology from the University of Stuttgart-Hohenheim, and during his postdoctoral research with Professor Günther Schulz in the Institute of Pharmacology at the Freie Universität Berlin, he was involved in the characterization of G-protein-mediated signal transduction processes. In 2000, he moved to RINA Limited, Berlin, as a senior scientist, working on the development and commercialization of novel solutions in the field of cell-free protein synthesis and labeling, and in collaboration with QIAGEN and Roche, he developed the novel eukaryotic in vitro translation system. Since 2010, he has led a large group at the Fraunhofer IBMT and became head of the department Cell-Free Bioproduction. His laboratory exploits cell-free protein synthesis as a versatile tool for functional genomics, for example, cell-free synthesis of antibodies, membrane proteins, glycoproteins, as Thomas already said. Stefan is also a lecturer at the Freie Universität Berlin, at the University of Applied Sciences Berlin, and of the University of Potsdam. And he is an affiliate of the Technical University of Berlin. In addition, even, Dr. Kubik is a member of the Faculty of Health Sciences, a joint faculty of the Brandenburg University of Technology in Cottbus-Senftenberg, the Brandenburg Medical School Theodor Fontane, and of the University of Potsdam. Recently, he founded the company B4 Pharmatech, a startup company on the campus of the Freie Universität Berlin, focusing on the production of bioorthogonal systems for protein labeling. There, he holds the position of a managing director. Stefan, it is a great pleasure to host you as our guest speaker. The stage is yours. We are looking forward to hearing about your research. And for the audience, please post your questions by using the Q&A chat of Microsoft Teams.
Yeah, Raimund, thank you very much for adding these details. It's my pleasure to introduce you into the field of cell-free bioproduction. In particular, I will focus on those proteins which are important for therapy, diagnostics, and several biotechnological applications. The company B4 Pharmatech is located in the southwestern part of Berlin, directly on the campus of the Freie Universität Berlin. And here we have a protein production.
Would you please share your presentation?
So I hope that you can see the presentation right now.
Here we go. Perfect. Thank you.
Sorry for that. So we'll directly jump in here. B4 Pharmatech is a company located in the southwestern part of Berlin, and it's directly on the campus of the Freie Universität Berlin. We have strong support from the Freie Universität Berlin, and we are in a clear interplay and discussion with all of the scientists on the campus, which are scientists from chemistry, but also biochemistry at the Freie Universität Berlin. The company offers a protein production service. Of course, we are also focusing on those proteins which are difficult to express, such as the membrane proteins. We produce antibodies and recombinant immunotoxins, which goes alongside with linkology that Thomas has already mentioned. Of course, we also produce enzymes, and sometimes enzymes are really difficult to express, in particular if they interfere with some intracellular components. Protein labeling is clear in the scope of our activities. Bioconjugates and the chemoselective protein modification is a prerequisite to add linker moieties, cargos, and so forth to a defined protein. So therefore, we need these bioorthogonal systems, which are constantly developed at the company B4 Pharmatech. In the end, for high throughput solutions, we need either on-chip automation or other types of high throughput assays, which are developed at B4 Pharmatech. And if you want to produce your protein of interest on your site, in your laboratory, we will be happy to offer you the cell-free systems and reagents. Now, why do we need cell-free protein synthesis? If you look at the left-hand side, you see this excellent in vivo protein production systems, and sometimes they work very nicely for the production of proteins. Prokaryotic systems are the systems of choice if we want to produce a protein with a very cheap, cost-effective manner, if it can be produced in a prokaryotic system, and if, in particular, the post-translational modifications are in this way that you can really apply this protein to subsequent assays and further applications. If you look at yeast, yeast is also an interesting host for protein production, but differs somehow in glycosylation from other eukaryotic systems. Insect cells, in particular those from Spodoptera frugiperda, those are the SF9 and SF21 cells. Those are the systems of choice for high-yield protein production, in particular also membrane proteins. And other mammalian cells, like CHO cells, are the classical cell line for the production of proteins in pharmaceutical and pharmacological processes. Now, all of these systems suffer of several disadvantages. In particular, if it comes to the point of the production of toxic proteins. So, for example, for the production of recombinant immune toxins, we need toxic proteins. On the other hand, for the development of several assays, we want to have the toxic protein as an active protein. So, overexpression of these toxic proteins is often impossible in any in vivo system. On the other hand, those proteins which are not really toxic, but the effect of the overexpression of the protein results in toxic effects to the living cell, can also not be produced in an in vivo system. So therefore, we need cell-free systems, and in this particular case, these cell-free systems have a clear standalone criteria for the production of toxic proteins. On the other hand, in vivo production processes are time-consuming processes, costly processes. So for the overexpression of an individual protein, it takes at least 24 hours. Whereas in cell-free system, within 60 minutes, a protein of interest can be produced. Now, how do we prepare these cell-free protein synthesis systems? Of course, we also start with a fermentation process, first of all. And as you can see, we typically grow these cells in suspension in chemically defined media. We do not use any animal components. So the process is free of these animal components, and we grow the cells in suspension to high densities, 6 times 10 to the power of 6 or 7 and even beyond. It's a repeated batch process or a perfusion process. We harvest the cells, we centrifuge the cells, we spin them down at 4 degrees, and we collect the cell pellet in a very mild procedure. So with a high density packed cells are then disrupted by a capillary-based procedure. And this so-called initial cell lysate or crude lysate is then further purified by chromatography, and we supplement an energy regeneration system to the cell-free system. At this particular point, the lysate can be stored at -20 or -80 degrees. So this is an excellent way of constant producing of a lysate, storage of a lysate, and shipping of a lysate. So this is why we are able to ship the cell-free system to your lab for the use on site for fresh production of proteins. So the protein production process itself is depicted here. We take the CHO lysate or insect lysate or eukaryotic lysate, which does not only harbor the ribosomes, but also initiation factors, translation factors, elongation factors, and of course the amino acyl tRNA synthetases, which pre-charge the tRNA in the cell-free system. We add either a linear template or a circular template, and in a coupled transcription translation process, the protein of interest is produced. And if you're working with membrane proteins or secreted proteins, membrane proteins are embedded in vesicles, and secreted proteins end up in the luminal part of these vesicles. There's a constant energy regeneration system. And as you can see, if you're working with the linear templates, such as PCR-derived templates, the entire process is devoid of any cloning steps. So we do not generate any genetically modified organisms, and therefore we do not even need an S1, an S2, or an S3 lab. Even viral or bacterial toxins and proteins can be produced in a fully automated manner without using an S1 lab. And even the linear templates do not need to be purified. You can directly pipette PCR templates into the cell-free reaction without prior purification. Now, in principle, the reaction mode are as follows. We can either produce the protein in a batch-based system, which is the classical Eppendorf tube or a 96-well plate, or if you want, you can produce your protein in a so-called CECF mode, which is a constant exchange cell-free system, where you have a chamber which is separated by a dialysis membrane. So the reaction chamber is the middle part, the feeding chamber is the outer part. Inhibitory byproducts are constantly removed, and ATP and energy is constantly spiked into the reaction chamber to ensure high yield of protein production. Now here's a direct comparison side by side. On the left-hand side, you see the classical in vivo pipeline. The in vivo pipeline starts with the PCR cloning, transfection, and selection of clones. Of course, you have to do fermentation and upscaling, cell disruption, and the isolation of the protein. This may take 1 to 2 weeks if you're really fast. I would rather say 1 to 2 months is more like the realistic time scale for the production of a protein in an in vivo system. In the cell-free system, as I already mentioned, you separate the production process of the lysate from the protein production process. So protein production itself takes only 60 minutes to 90 minutes. The overall process, if you calculate the production of the lysate of the cell-free system, plus the production of the protein, is completely finished within 1 to 2 days. Of course, we are also able to produce some complex proteins in the cell-free system, as you see on the right-hand side. The longest time may be the time it takes until the PCR primers arrive, and then amplification of your protein of interest and production of the protein of interest is dramatically fast. Now, as I mentioned, we produce some specific templates by linear amplification. In a two-step procedure, in the so-called expression PCR procedure, we first of all amplify the gene of interest by shorter internal primers. We use then the longer outer primers to add some specific sequence motifs to this PCR product. So what type of DNA is added to the gene of interest? As depicted here on the left-hand side, we add an internal ribosome entry site. And this is the internal ribosome entry site of the cricket paralysis virus, which is an iris site that does not depend on any other factors. So it's a factor-independent iris site mimicking a tRNA, which ensures the initiation of protein synthesis on the ribosome. So therefore, we do not need any cap component by using this typical iris site. Capped messenger RNA may also be used in eukaryotic cell-free systems. And if you want, you can also add some internal PCR primers, thereby introducing point mutations, for example, stop codons like the AMBER stop codon for site-specific labeling of a protein. So this simple procedure is then 2 or 3-step PCR for the introduction of the AMBER stop codon at a defined position for the modification of your protein of interest at a very defined position. So now we can spike in the DNA, a circular plasmid DNA, as long as it harbors a T7 promoter. Might also be another promoter if we add another polymerase, the linear PCR product to the so-called coupled system. Transcription by T7 RNA polymerase leads to RNA. RNA is then simultaneously translated into the protein of interest. Unfortunately, the protein of interest doesn't jump out of the reaction. This we have to optimize, but the protein of interest can be easily purified directly from the reaction. On the other hand, if you do not want to work with a coupled system and you want to avoid any polymerase, of course, RNA, either capped RNA or non-capped RNA, can be directly spiked into the reaction. In this dialysis mode, the so-called continuous exchange cell-free system, byproducts are constantly removed. This prolongs the reaction time up to 24 or even 48 hours, leading to high yield production of a protein, and ATP and GTP is replenished to a 10 kD membrane. Now, as we're working in an open system, the system can be fully automated. And as shown here, you see the blue part of this robot. This is the cooling rack at 4 degrees. 96 well plate is over here. Lysate is pipetted into these individual wells, and then individual templates are applied to the 96 well plate. Grabber takes the plate, puts it into the incubation oven, which is over here. Sorry for the black over black, but can hardly be seen, but the incubation oven is here. And then 96 proteins can be produced in parallel within 60 minutes. Here we see a typical time course of protein production in such a fully automated system. Now, this is an essential prerequisite, for example, for the design of enzymes, mutational analysis of enzymes. Artificial intelligence in combination with enzyme design is an excellent application for such a fully automated production of enzymes in this robot. Now, we are devoid of the production of any genetically modified organisms as long as we do not use a plasmid prep prior to the cell-free protein synthesis and as we directly spike in the DNA. The system is scalable. You can even start below microliter volumes for on-chip protein synthesis, and you see the publications up to the range of 100 liters. 100-liter range is then a wave back where you incubate the entire reaction in 100-liter plastic bags. As the system is an open system, we're able to add chaperones, detergents, and so-called compatible solutants, so all different types of substances increasing, for example, the solubility of a given protein. The system enables a rapid domain screening. You can even chop down your protein of interest by amplifying the particular parts from the gene, the encoding sequence, and thereby expressing several subdomains. Even if the subdomain is constantly active, it will be produced in the cell-free system and is not affected by any constraints in vivo protein production. Labeled proteins, protein conjugates can be produced in parallel, so thereby you're able to detect the optimum amper position for labeling of your protein of interest. We have high yields in both, I would say, pro and eukaryotes, and some post-translational modifications are possible in eukaryotic cell-free systems. So here's an example taken from a prokaryotic cell-free system. We produced a protein and we modified several parameters. Sorry for the German here, I will explain that in detail. As you're working in an open system, you can even play around with the pH, which is often impossible in an in vivo system. And by shifting pH to different concentrations here, the solubility of the protein is dramatically increased. Functionality and solubility of the protein can be optimized by changing the production temperature, which is also impossible in an in vivo system, but in a cell-free system, this can be done easily. You can add these so-called compatible solutants, which are, say, mild detergents, such as Bridge, for example, or DDM or other detergents. You can play around with the ion concentrations. Even the cell-free system itself can be diluted to reduce the speed of protein production and thereby increase folding of a protein. And this way, by combining all of these different types of optimization possibilities, you end up with this robot-made automatization of soluble protein production. Now, what is the advantage of a eukaryotic cell-free system? As I already mentioned, post-translational modifications. So for these post-translational modifications, we need the subcellular compartments, such as the endoplasmatic reticulum. So for a specific lysate preparation out of these eukaryotic cells, which have been grown in the fermenters, remove the outer cell wall, we remove the nucleus, and we revesiculate the endoplasmatic reticulum. So we have to keep the functional integrity of these subcellular components as intact as possible. So the entire lysate production procedure differs dramatically from the prokaryotic lysate production procedure. And as a result, we end up with these types of so-called ER bodies. This is a high magnification of the eukaryotic cell-free system, and you see this is what happens to the endoplasmatic reticulum. It ends up in these type of vesicles with a typical diameter of 1 micrometer to 10 micrometers. So there is an ER present in the eukaryotic cell-free system. Now this can be combined with both of the systems. In the batch-based system, we can use these vesicle-based systems from eukaryotes, but also in the dialysis system, of course, we have to avoid any clogging effects on the membrane. So as a result, if you produce a protein of interest, once the protein harbors a signal sequence, it will be translocated through an active translocon into the lumen of these ER-derived vesicles. Membrane proteins will be embedded throughout the translocon into the membrane of these vesicles. So this ensures that 100% of the membrane proteins that end up in this vesicular structure have the same orientation. So it's not a statistical incorporation of a membrane protein in a lipid bilayer, but it's rather an oriented, directed introduction of the protein by the translocon into the membrane of the vesicle. Now this on the upper right-hand side is the technical application, which can also be operated in any automatic surrounding in high throughput robots. Here you have the reaction chamber, here's the dialysis membrane, here's the feeding chamber. Within the reaction chamber, the protein of interest is produced, and then you handle 48 chambers on one plate in a fully automated manner. Now, if you want to reduce the volume due to pre-screening processes, for example, you can use cell-free proteins since this is directly on a chip-based platform. We have published this in Lab on Chips some years ago. Here you see the transcription compartment, which was developed with the Fraunhofer Institute in Berlin. In the transcription compartment, RNA is constantly produced, binds to particles. Particles are transported by this magnetic mover, which is below the chip surface. Here, these particles are washed and the RNA is constantly spiked into the cell-free protein reaction, which is in this chamber. The entire construct can be stored together with the lysates, and if you need your protein of interest, it's right on time production of this particular protein. So-called TRIT platform then leads to the production of the protein, which can subsequently be spotted on chip surface or even be used directly after this procedure. Now, these vesicles that are already depicted in the eukaryotic system, they can be addressed even on these chip-based platforms. And as you see on the left-hand side, typical reporter genes as YFP fluorescent proteins here produced in the cell-free system. Once you add the signal sequence, such as the melitine signal sequence, the protein of interest is translocated into the lumen of these ER-derived vesicles, as can be seen on the right-hand side. Vesicles are rock stable, can be stored at -20 or -80 degrees, can be pipetted on chip surfaces, even immobilized on chip surfaces, and then can be decorated by the protein of interest. Even several membrane proteins can be produced and immobilized in these vesicles in a subsequent way or in a parallel way. Now, what about post-translational modifications in these cell-free systems? First of all, glycosylation. Is there any type of glycosylation possible in these systems? It is, and as you can see, here is the result of the eukaryotic cell-free system. This is an autoradiograph, so for labeling, we spike in 14C labeled leucine. Leucine is incorporated into the cell-free produced protein, so you do not necessarily need any Western blotting. You can directly visualize the protein of interest in this autoradiograph. And as you can see, the upper band depicts the glycosylated EPO, and upon deglycosylation when using PNGASF or N2H, this band drops down to the bands with a lower molecular weight, thereby indicating that there is glycosylation in the cell-free system plus signal peptide cleavage. So we are able to produce a glycosylated protein. What type of glycosylation can we expect in these eukaryotic cell-free systems? We analyzed that in detail by mass spectrometry, and this was published by Annette Zemella in 2018. We see these different types of glycomoieties which are attached to all three N-glycosylation positions in the protein. So the clear advantage is that all of the end glycosylation positions are addressed. You find the glycomoieties in all of these positions, and these are the types of glycosylations which we see in the self-reproduced EPO. Now, as we're working in a cell-free eukaryotic system, we can directly pipette the result of our protein synthesis onto cell culture. And as you see on the right-hand side, this is the cell culture where we directly pipetted the self-reproduced EPO into the medium of the cell culture, and we analyzed the growth of these cells. So this again dramatically speeds up the entire procedure. No purification is necessary for generating EPO, which is applicable to any cell culture applications. As you see, there is a growth curve, so cell-free produced EPO induces cell growth. It's active and it's actively produced in the lysate. Now, what about further post-translational modifications and other downstream applications for cell-free protein synthesis? Here you see a single molecule analysis where we used EYFP harboring a CARX box. So those proteins harboring a CARX box are typically lipid modified. So by lipid modification, these proteins are attached to the lipid moiety unless EYFP would be soluble. So here you see that this protein clearly follows the membrane. The inset here is a single molecule analysis, which was published with our partners, lab of Thomas Schmidt in the Netherlands. So the production of lipid-modified proteins is possible in this cell-free system, and even fluorescently labeled proteins for single molecule analysis and for the detection of protein-protein interaction on a molecular level can be facilitated by cell-free protein synthesis. Now, the largest class of membrane proteins, which is clearly in the scope of several groups and research and development, but also in pharma screenings, are the G-protein coupled receptors. And as we know, sometimes G-protein coupled receptors are really difficult to express, in particular if they interfere with signal transduction pathways inside of a living cell. So the method of choice when using cell-free protein synthesis systems is the production of the G-protein coupled receptor in the so-called batch mode, simple Eppendorf tube. Translocate the GPCR into the previously mentioned microsomal membranes, spin down these microsomal membranes with the GPCRs in a defined orientation, and then apply any downstream analysis, such as ligand binding, or interaction studies, or Brett assays, Fred assays, whatever. So here's the result of a cell-free production of a G-protein coupled receptor. Typical example here is the endothelin B receptor. We've used the endothelin B receptor to GFP at the very C-terminal part, so fluorescence ensures the production of the full-length protein. And as you can see, the membrane protein is embedded in these membranes in the eukaryotic cell-free system. Membranes can be mobilized on the surface, can be collected, and thereby you can concentrate the G-protein coupled receptor for any downstream applications. So here's the typical application: ligand binding, screening of a ligand, and analysis of receptor-ligand interaction. Of course, first of all, we have to take into consideration for these experiments that the vesicle is closed in the opposite orientation in comparison to a living cell. So we have to bring in the ligand to the luminal part of a vesicle. Otherwise, there would not be any ligand binding. So this clearly differs from a cell surface. On the cell surface, we have the GPCR, G proteins in the membrane. Ligand comes from the outer side, activates the GPCR, and signal transduction is activated into the luminal part of the living cell. Here, vesicle is closed in this orientation, so ligand has to apply to the luminal part. On the left-hand side, you see a typical chip-based analysis of ligand binding to a G-protein coupled receptor. First of all, left-hand column here is the endothelin B receptor, which is expressed in the vesicles and immobilized on the surface. In this column here, you see the fluorescently labeled ligand, which is in Psi3 labeled endothelin 1. And initially, upon application of the ligand, you see no ligand binding. And this is the control experiment that clearly depicts that all of the GPCRs have one orientation. They show the typical orientation with the ligand binding site to the luminal part of these vesicles. So upon using a mild detergent, Bridge 35, at a very low concentration, vesicles perforated but not destroyed, and in this way the Sil3 labeled ligand has open access to the ligand binding site of the GPCR, and you see the ligand binding directly on chip. So this whole procedure takes 60 minutes of production of the GPCR, 1 minute for a ligand binding experiment. So it's dramatically fast in comparison to any in vivo system, which takes 24 hours or even beyond, only for the production of the protein. Now, what about multimeric membrane proteins? Here you see KCSA, a tetrameric ion channel, which was produced in the cell-free system in collaboration with the Fraunhofer ICBB and Srushan and Donda party. Here you see the tetramer in the gel and is already assembled in the cell-free system. Now, the functional analysis of these ion channels is done in collaboration with the company Nanion, I would say the world market leader in electrophysiology. And here you see we can directly apply these vesicles to electrophysiological analysis of ion channels. The cell-free produced KCSA was pipetted on a lipid bilayer in this Orbit 16 machine. You see a fusion event and immediately followed by the activity of the cell-free produced multimeric ion channel. Now here is the combination of a cytotoxic protein, the thermostable direct hemolysin, which is the toxic moiety in Vibrio parahemolyticus, unfortunately currently in the Baltic Sea. This thermostable direct hemolysin can be produced in the cell-free system, ends up in a tetramer, dimer, and monomer, and upon fusion, the tetramer shows electrophysiological activity, and this was published meanwhile, these results. So you see the cell-free production of even multimeric proteins is possible in eukaryotic cell-free systems. Now, of course, these TDH can also be analyzed directly on the typical blood agar plates for the activity assay of this cytotoxic protein. And here we played around with different tags and different positions, either N-terminally tagged protein or C-terminal tagged proteins and other tags to optimize the activity plus purification options of this toxic protein. And as you can see, these four varieties of the protein can be produced in an active tagged manner. Now, as I've been explaining so far, we produce the proteins based on 20 canonical amino acids, which are typically pre-charged. So the amino acyl tRNA synthetase pre-charges the tRNA and are then subsequently introduced into the protein of interest on the ribosome. Now, is there a chance to introduce non-canonical amino acids? Of course. Here is another example of pre-charging a tRNA. The right-hand side was already explained, enzymatic pre-charging of a tRNA. On the left-hand side, you see the chemical charging of a cognate tRNA. We simply have the chemical moiety, 2 nucleotides are added to the chemical moiety, and an RNA ligase then ligates the tRNA minus 2 nucleotides to the chemical moiety plus the 2 nucleotides. So this is a simple RNA ligation step, which was already published a few years ago by Ned Budisha and others. And as a result, after appropriate folding of the tRNA, the pre-charged tRNA can directly pipette it into the cell-free system for further protein synthesis. Now, the pre-charged tRNA can then either be used for statistical labeling if the anticodon is, for example, a phenylalanine anticodon or any other anticodon. Here are some examples. On the left-hand side, you see the introduction of biocytin. So the biotin moiety was introduced in the protein, and here even a glycosylated protein could be produced, and the biocytin was introduced into the de novo synthesized protein, as after blotting, the protein was detected by the streptavidin peroxidase. So this is a kind of a Western blot without using any antibody. And the introduction of the biotin moiety, of course, has a lot of downstream applications such as immobilization, purification, and as mentioned here, detection of the protein of interest. On the other hand, we introduce fluorescent moieties such as the body pyTMR lysine. And as you can see here, fluorescently labeled proteins can be directly detected in gel, even without drying the gel or even without blotting the gel. Sensitivity is in the same range here as compared to the autoradiogram, and I would say the bands are even clearer and can be detected easily and straightforward here in this fluorescent-based protein labeling assay. Now, in cell-free systems, there are lots of options for the introduction of non-canonical amino acids. Mainly modified tyrosines or phenylalanines are used for the incorporation of non-canonical amino acids into a defined protein. Of course, photoreactive groups are possible, metal chelators, for example, redox active side chains, and so forth. I showed the results for the body pyTMR, fluorescent labeled, and the biocytin for the introduction of the biotin moiety. Now, how can we generate some biosensors based on the self-reproduction of G protein coupled receptors? We published this with Annette Zemella, and here is the example of the production of the adenosine A2A receptor, which was fused to nanoluciferase. So nanoluciferase is the type of luciferase that can be simply added to the C-terminal part of a given protein. So now the fluorophore is introduced site-specifically at a given position within the protein, here internal loop 3. And this results in an excellent Brett assay. Activation of nanoluciferase leads to the excitation of the emission here of the light from the fluorophore. And upon ligand binding, we see a clear shift of this excitation. So the production of such biosensors is possible in cell-free systems. Now, what about introduction of site-specific of amino acid at site-specific positions? As I already mentioned, with the expression PCR, we're able to introduce the AMBA stop codon at a defined position within the given messenger RNA, within the given DNA, and then the transcribed messenger RNA harbors the AMBA stop codon. You may then add the pre-charged tRNA, or you may use an enzymatic pre-charging of the tRNA, which is then the carrier to bring in this chemically modified amino acid in this particular position within a given protein. Now here we need an orthogonal system. So what does this mean, an orthogonal system? Of course, we have the endogenous synthetases that pre-charge the endogenous tRNA. And this is the process which we need for protein production. This has to be constantly active in the cell-free system. But if you're working with the background of a eukaryotic cell-free system, we may spike in components from a prokaryotic system or from archaea. And we might use the orthogonal synthetase from prokaryotes or from archaea to pre-charge the orthogonal tRNA. So those two components are spiked into the open cell-free system. In this way, we're able to pre-charge this particular tRNA designed against the AMBA stop codon, and in this way we end up in a suppression product which is site-specifically labeled. We prefer the introduction of para-aceto-L-phenylalanine, as para-aceto-L-phenylalanine can be easily modified by Staudinger ligation, kappa-catalyzed cycloaddition, or even strain-promoted cycloaddition in a kappa-free procedure for the production of site-specifically chemically modified proteins. Now, here's one example of such a bioorthogonal system. We use the E. coli tRNA tyrosyl tRNA synthetase for the introduction of the non-canonical amino acid. Of course, to ensure the bulky substrate to enter the active site of the enzyme, there are some mutations necessary in the enzyme. And on the other hand, also tRNA needs to be modified slightly to ensure optimum pre-charging of the tRNA with this enzyme. So in this way, for example, the propagyloxy-L-phenylalanine can be pre-charged to a given tRNA. On the other hand, same enzyme also pre-charges the tRNA with para-acido-L-phenylalanine. There's a second enzyme for methanosarcina masei. It's the tRNA pyrolysyl tRNA synthetase, which pre-charges the tRNA with pyrrolysine, cyclooctene lysine, SCO, for example, TCO, and other amino acids. So these non-canonical amino acids can then be introduced in your protein when using this enzyme for pre-charging of the given tRNA. Now, in the eukaryotic system, these amino acyl tRNA synthetases can even be engineered into the genome by using a CRISPR-Cas technology. So in this way, the amino acyl tRNA synthase is already produced in the living cell, and the lysate prepared from this living cell already harbors the amino acyl tRNA synthetase. So this is a very user-friendly, convenient way of producing labeled proteins, as you simply have to spike in the non-canonical amino acid plus the tRNA. And the template, which harbors the AMPA stop codon. So in this way, you end up in a site-specifically modified protein harboring the ACID group for further chemical modification. Now comes the combination of all of these procedures when producing antibodies. Of course, in these cell-free systems based on CHO lysates or insect lysates, antibodies can be produced, IgGs, all different types of single chains, nanobodies, and so forth. And this simply depends on the addition of the template. We add the heavy chain, but also the light chain, and this was already published, to assemble the IgG, and we introduce the AMPA stop codon into the heavy chain at a very defined position. In the end, body-pide hemolysin tRNA was added to visualize the different types of antibodies that have been produced in the cell-free system. And this body-pide label is introduced site-specifically at the AMPA position 134. Now, for antibody production, we want to introduce the para-acido-L-phenylalanine at the defined position. The result shown here is the control experiment. Again, we labeled the antibodies with para-acido-L-phenylalanine in this case, and here we added the dilite 650 to visualize the different types of different types of antibodies that have been produced in this system. So to ensure the activity of these antibodies, we typically use SPR-based experiments. And as you can see, here is an antibody produced in a cell-free system designed against the phosphorylated SMAD protein, which was published in collaboration with the Fraunhofer ICBB, where we see the binding of the antibody and even in the presence of the label, and in the presence of a cargo, same experiment can be used for control. Now, what do we add to this para-acido-L-phenylalanine? Of course, here we have an excellent linkerology from the company Iris. Antibody produced in the cell-free system or any other protein is labeled site-specifically with para-acido-L-phenylalanine. Conjugation is done with this particular linkers, and some of these linkers are optimized. They can be eliminated in a traceless part, so thereby the payload is released in an intracellular part from the self-reproduced site-specifically labeled protein. Now, the excellent combination of cell-free protein synthesis and linkerology is depicted in this cartoon. Here you see the valyl-citrulyl dipeptide fragment, which was attached to the para-acido-L-phenylalanine, which directs the drug to the self-reproduced protein. And after the cleavage by cathepsin B, the intracellular cleavage, this part is removed, which leads to 1.6 elimination, and the drug is released internally in the site, inside of the living cell, without any trace of a linker, and the combination of high throughput production of different types of antibodies plus different types of linkers plus different types of drugs leads to a complete new pipeline for the production of antibody drug conjugates. As you can see here, IRIS also offers these different types of linkers, which show a different time frame of release of the drug. This is lowest version, faster, and even here the fastest version of drug release. So this can be then applied in a very controlled manner when combining cell-free protein synthesis with linkerology. So this depicts the entire pipeline of antibody drug production when using cell-free systems. First of all, we produce the different formats of antibodies. We add the para-acetoalpha-phenylalanine, we add the linkers, we conjugate the payloads, and we directly apply the resulting constructs to the high throughput analysis of antibody drug conjugates on cell cultures. Again, the advantage, we're working with a eukaryotic cell-free system, no endotoxins are present, and the constructs can be directly applied to cell culture as there's no carryover of any toxic moieties resulting from E. coli, for example. With this, I would like to thank you for listening. I would also like to thank our partners from academia, in particular Fraunhofer ICBB, Annette Zemella, Sushan Dondapadi, Franziska Ram, and also from the Technical University, and in particular the group of Rainer Haag and Rainer Haag working here at the Freie Universität Berlin, our partners in Potsdam, the BTU Cottbus-Senftenberg, and in Leipzig. So with this, I would like to thank you again for listening, and please have a look at our webpage, which clearly depicts different applications of cell-free protein synthesis. Thank you very much.
Yeah, thank you so much, Stefan, for sharing your results with us. And during your talk, we collected quite some questions. Very first one was about, say, the typical protein yields in cell-free production systems. I saw on one of your slides up to 600 micrograms per milliliters in a titer plate. Is this also realistic for bulk scale, say up to liter scale, or what are their typical yields?
Yeah, thank you very much for the question. As we're working in an open cell-free system, there's linear scalability. And the linear scalability, this means that we are able to optimize the reaction in a sub-microliter volume and then scale up the reaction to the 100-liter volume, even by harboring the same reaction conditions and the same yields. So yield is typically not affected by scaling up the reaction. So a typical yield for the batch-based system is in the range of 100 microgram to 700 microgram roundabout, depends on the prokaryotic or eukaryotic cell-free system. And in the dialysis mode, we end up beyond 1,000 microgram per milliliter. So this yield is even higher than in any living cell, as we focus the entire protein production machinery on only one template. So we do not have to produce in parallel all of those different housekeeping genes. We simply produce one protein of interest in the cell-free system.
Okay, thank you. Then one question, because you have to prepare those lysates. Janis Beutel asks, does Cullen often plug during purification of those lysates, or do you pre-filter them, or what is the typical procedure?
The procedure, of course, it differs for the prokaryotic and for the eukaryotic cell-free system. We use special columns that do not show any plugging effects. So, of course, we're working with a hydrophobic mixture, so we have to take this into consideration. Hydrophobic components, but also vesicles, have to run through the column. That's correct.
Yeah. So, well, since you have to do some work before in batch and bulk production, is there a kind of a thumb rule by which factor is a cell-free batch production more expensive to achieve the same yield compared to a classical in vivo expression?
Well, as I already depicted, there's a clear standalone criteria for cell-free protein synthesis. Once you end up with a protein that cannot be produced in a living cell, or where the protein is misfolding, inactive or whatsoever, then the cell-free system is the system of choice. Of course, that's a standalone criteria. And a lot of poor forming proteins, for example, which are of highest importance also for technical applications, such as high throughput sequencing, they can only be produced in the cell-free systems in parallel in a high amount. So it depends. I mean, you have to take into considerations that the time-saving effect is also has also been calculated. In the cell-free system, we need 60 minutes. You can produce that in a fully automated way, 24 hours by a robot, which makes the system quite cheaper than an in vivo system.
Okay. So one more question from Yannis Beutel. I hope he doesn't want to use it for doping purposes regarding the glycosylation pattern in EPO. So as far as I know, also some doping tests use these glycosylation patterns to realize whether this is external or internal. So is the glycosylation of EPO achieved in cell-free expression the same as when producing it endogenously?
The glycosylation differs, as I have depicted the results from mass spectrometry. We have this type of core glycosylation plus trimming on the level of the endoplasmatic reticulum, and this leads to the active EPO. I can assure you that as far as we have contact with those persons that are responsible for doping controls, this type of EPO is also detected as doping.
I see. Very good. So one more question. Do you use off-the-shelf cell-free systems sometimes, or do you create them all yourself on your own lysates?
We produce them on our own self, as I mentioned, by fermentation of these cells, CHO cells, insect cells. I prefer insect SF21 plus CHO cells. Those two cell types are those for the eukaryotic lysates. And as the fermenters are typical 5 to 30 liter fermenters, we're then capable of producing large materials and bulk materials of cell-free systems, which ends up then typically in a volume of 25% lysate within the final reaction of the cell-free system. And the typical volume for the cell-free system is the 50 microliter volume to optimize reaction conditions.
Okay. Something completely different. You showed the statistical introduction of bulky fluorophosphamide, not so bulky, but polypi with lysine within the lysine side chain. Does that, especially the statistic introduction, interfere with the protein structure folding?
Yeah, this might interfere with the protein structure, of course. You're absolutely right. Yes. So statistical labeling is rather for controlling the protein expression per se by in-shell analysis. The simple procedure, you take the STS shell, you load the STS shell, wash it in methanol water, and pour it on a typical UV screen that is used for RNA or DNA analysis. So it's a very simple and fast procedure without blotting. No Western blot is necessary, no antibody, simple for the detection of the protein. To ensure the maximum functionality, I would always prefer the site-specific labeling. And site-specific labeling with the para-acetyl-L-phenylalanine plus conjugation of fluorescent moieties. So it's typical bioconjugate, which is then formed, and this should not affect the functionality if the label is outside of an active enzyme, of the active center within an enzyme.
Yeah, and here we go already with then the site-specific labeling. The next question is there a significant percentage of incomplete stopped sequences when introducing non-canonical amino acids via an AMBA suppression?
This is a very, very interesting question. Yeah, it's a context-dependent introduction of the non-canonical amino acids. So some of these flanking nucleotides, flanking amino acids in the sequence, they typically affect then the labeling. So we as the experts are happy to guide you through the labeling of your protein. You may also send us an email with the sequence of your protein of interest, and we suggest the optimum positions for site-specific labeling. And on the other hand, we have to take into consideration that there is also a stop codon at the very end, at the 3' end of the DNA or RNA. So here I always suggest to add 2 or 3 stop codons, of course not the AMBER stop codon, in all of these 3 reading frames to ensure appropriate termination of protein synthesis.
Okay, and something in a similar direction, can one introduce several different non-canonical amino acids into the same protein sequence?
Yes, in principle that's doable. There are also the publications where you enlarge the triplet code to 4 nucleotides. So by enlarging this triplet code to these 4 nucleotides, you can then use 2 different 4 nucleotide constructs to introduce 2 different labels into a protein of interest. It's somewhat bit more difficult, reduces the overall yield of labeled protein, double labeled protein, typically below 10%. So,
and I see there were some questions coming in recently. The one is, can you specify which separation you use and what you fractionate for? So I guess for the lysate, for the making the lysates.
Yeah, for making the lysates, we use a capillary-based system just to remove the outer cell wall. Fractionation is done in such a way that we have to remove the nucleus and cell debris. Of course, this is classical standard procedure. But the supernatant already harbors the vesicles of the ER. So this depends on buffer conditions and the initial procedure for preparation of these lysates.
Mm-hmm.
And this allows the user to later on separate the self-reproduced membrane protein, which is inside or embedded in the vesicle, from the rest of the cell-free protein synthesis reaction by a simple 10 minutes, 16,000 G centrifugation step.
Okay, and then one question a bit also to the beginning of your talk. You mentioned even liter scale reactions up to 100 liters. Can you disclose what protein or what kinds of proteins you did produce at such a big scale?
You see the publications from Zawada et al. There are also other groups working in this field, and they produce growth factors in the 100-liter range.
Mm-hmm. So indeed something also of commercial interest. Yeah.
Exactly.
Okay. Yeah, so thank you for disclosing this. I see currently no further questions. So again, if you have any questions left, do not hesitate to contact us or also contact Stefan Kubik directly. He has shown you the link of his brand new webpage. So you can also see, I guess, the link in the chat right now. Exactly. Klaus has published this, so it's www.b4pt.de. With this, we would like to close our online workshop for today. If you are interested in using the capabilities for cell-free protein production, do not hesitate to contact us or to contact Stefan directly. As a follow-up, we will provide the recordings of this workshop. And thanks again for joining us. Thanks to both of our speakers, and also a big thank you to our QA manager, Klaus Helm, for taking care of the technical support. We hope you could collect some helpful ideas and information from this workshop, and you make some interesting experiences with this elegant technology of cell-free protein production. We are looking forward to meeting you in the next workshop in December, which will be announced on time on our homepage and via social media. So hopefully enjoy a beautiful autumn, stay healthy, and see you soon. Goodbye.